Welcome to Medical Concept. Today topic is about PCR and gene therapy. Uses of PCR. Used to amplify DNA gene in synthesis of cDNA and in DNA sequencing. Identification of criminals by DNA fingerprint. That is the uses of PCR. And to recognize them caused by viral and bacterial infections like malaria parasite and HIV and tuberculosis, to diagnose cancer, and in prenatal and antenatal genetic diagnosis for gene mutation, sickle cell disease, and phenylketonuria. And types of PCR. C. Conventional DNA based PCR, reverse transcription PCR, asymmetric PCR, Anchored PCR, Inverse PCR, Nested PCR, RT-PCR, Real-Time PCR, and rapid amplification of cDNA ends, that is called RACE, and rapid random amplified polymorphism DNA. Now coming to the reverse transcriptase PCR, used to amplify, isolate, or identify to known sequence for a cell or tissue RNA library. mRNA is deducted from living organism. PCR preceded by reverse transcription, widely used in expression mapping. And 10th polymerase for thermophilus is used. This enzyme has both DNA polymorphase activity and RTS activities at a high temperature. This allows both cDNA synthesis from mRNA followed by PCR amplification. Presence of HIV RNA in blood can be detected as early as 4 weeks after infection. And multiplex PCR, by targeting multiple genes at once, additional information may be elicited from a single test run that otherwise requires several times the reagent and time of perform. And coming to the next, gene therapy. Now we are going to the topics here: types, approaches, delivery systems, techniques, hurdles in gene therapy. First, what is gene therapy? Everyone knows what is gene therapy. Technique for correcting defective genes that are responsible for disease. Okay, that is called gene therapy. And somatic gene therapy, it have some types. Okay, so now somatic gene therapy, gene material is transferred to the body cell, example liver cell. Okay, affects only the target body cells in the patient and is not passed to future generations. Short lived because the cells of most tissue ultimately die and replaced by new cells. And gene line gene therapy, gene material is transferred to either sperm or egg, which is transmitted to the successive generation, results in permanent changes. Next, potent for offering a permanent gene therapeutic effect for all who inherit the target gene. Possibility of eliminating some disease from a particular family. And now coming to the approaches: replacing, inactivating, introducing a new gene, replacing a mutated gene that causes disease with a healthy copy of the gene. Inactivating or knocking out a mutated gene that is functioning improperly. Introducing a new gene into the body to help fight the disease. And delivery systems. Delivery systems are what are all plasmid maybe or viral vectors and transfers through the liposome, artificial chromosome vectors and receptor mediated endocytosis and antisense therapy. Now coming to the technique, okay. Ex vivo gene transfer and in vivo gene transfer. In ex vivo gene transfer, the cells with genetic defect from a patient are isolated or growth in culture. The genetic material is first transferred into the cells grown in vitro. Control process. Genetically altered cells are selected and expanded by many manipulations. Cells are the returned back to the patient. Okay. In vivo gene transfer, the genetic material is transferred directly into the body of the patient. More or less random process, small ability to control, less manipulation are needed. This is the only available option for tissues that cannot be grown in vitro, or if grown cells cannot be transferred back. Now coming to some disease. Severe combined immune deficiency, attachment muscular dystrophy, cystic fibrosis, familial hypercholesteremia, hemophilia, cancer, and Leber's hereditary optic neuropathy. These are all diseases that is used to gene transfer here. Okay, that's it, guys. Thank you.